Bismillah. Now we continue on the series of lectures concerning the course applied pharmacognosy designed for fifth year pharmacy student. The title of this second lecture is General Phytochemical Screening of Medicinal Plant Extract. At the beginning, general phytochemical screening means it's a process of identifying the presence or absence of secondary metabolite in the plant extract using cost-effective tests or assays. As a process, general phytochemical screening involves four basic steps. First of all, extraction and preparation means use of appropriate solvent to extract the plant active constituent, and preparations means use a suitable method for filtering or clarifying this extract. And the third and fourth sequential step is analyzing and interpretation. Analyzing means use suitable screening methods or analyzing methods, maybe chemical or physical methods, and interpretation means how to interpret these results obtained by using these methods. In general, the principle of phytochemical screening involves change in color or formation of precipitates. And at the end, the finding of phytochemical screening can be reported as plus or minus or presence or absence as simple as that. There are several methods, as I said before, there are several methods used in phytochemical screening. In this lecture, we will talk about the phytochemical screening for 13 important classes of secondary metabolites, and also we define the type of secondary metabolite that present in medicinal plant, and which have important therapeutic potential. As brief introduction, general phytochemical screening is a process of identifying the presence of various bioactive compounds or secondary metabolite in plant material by using cost-effective or appropriate tests. And the main purpose of phytochemical screening is to expel or to express or to understand the potential therapeutic properties of the plant. There are several plant bioactive components or secondary metabolites. As we know, secondary metabolite is a compound or chemical compound produced by plant but are not essential for growth and reproduction, but they have another clinical function for plants. They may be uh, attract pollinators or repel or defend or the plant against predators like fungi, insect, mold, and so on. So the important class of secondary metabolites having ecological function for plant also can be used therapeutically for treatment of many diseases. This compound includes alkaloids, glycosides, flavonoids, tannins, besides fixed oil and carbohydrate, although they are considered primary metabolites, but some of them have important physiological function for plant, especially they are source of energy such as carbohydrates and fixed oil. While protein also important, they are essential for growth and for action of many enzymes and also for immune system for the plant. Particularly screening for plant extract to detect the presence of secondary metabolite involves several sequential steps. Before we end with the result is the secondary metabolite is present or not, There are four sequential steps can be followed to determine the presence of the secondary metabolites. First this step is extraction. We start with extraction using solvent and suitable solvent according to the intended compound to be extracted, followed by preparation. Preparation means use of suitable filtration method to obtain or to separate the active fraction from inactive or from the plant mark, followed by interpretation and analysis. This will be interchangeable. Analysis means the use of suitable testing methods or screening methods, and interpretation means interpretation or explanation of the result obtained. In general, for phytochemical screening, the results or the interpretation of the result based on the change in color or formation of precipitate. There are several methods used for screening of plant extracts to detect the presence or absence of phytochemicals, and the main feature or the similar feature for all the chemical methods or all the methods used for phytochemical screening, they should be cost effective. They are feasible and with suitable cost to be carried out, because it is mainly used to detect if the secondary metabolite is present or not. And generally, in the findings, the result can be reported as positive plus if the secondary metabolite present, or absent, and you can write minus or negative signs for compound is not present in the extract. In this slide, there is several chemical screening methods that can be used for crude extract to detect the presence of secondary metabolite. Here, there is more than 11 secondary metabolite, 4 secondary metabolite with the suitable test used for detection. We start with alkaloid. The suitable or the most appropriate test can be used as general screening tools using dragon doors, Myers, Haggard's, Wagner's reagents. These are four types of reagents or tests can be applied to the presence of alkaloid. The dragon doors reagents, Meyer reagents, Haggard reagents, Wagner reagents, or Wagner test. This is used for phytochemical screening for alkaloids. For tannins, we used two methods: chloride test or polymer test. Polymer test or chloride can be used for tannins. And anthraquinones can be detected by using Bontrager test. Bontrager test. And flavonoid can be tested by Shinoda test or using lead acetate test. Glycoside tested by Wheeling reagent, Wheeling test, and by glacier acetic acid test. While cardiac glycoside can be tested by using Kilaklia test, and terpenoid steroid can be tested by sulfuric acid test. Saponins, which have the main feature, they have forming form, can be used by simple physical test by forming form test, which involve just shaking and sometimes heating of water extract. Fixed oil also can be tested by spot test, also considered as physical test. Just we use filter paper and we put a drop of fixed oil. If it persists for a long time, it could this may be signs it is fixed oil. Otherwise, can be what? Can be volatile, which is unstable at room temperature. Amino acid and protein can be detected by anhydride test and copper sulfate test. Terpenes and sterol can be tested by Lieberman Borchardt reagents or test. And phenols, lastly, can be tested by phenol test. In next slide, we speak in details or describe the details of these reagents, how can be performed, what is the suitable solvent for extraction, the preparation method suitable, the method of interpretation and method of analysis can be discussed in details. For testing the presence of alkaloid in plant extract, we use the four reagents for detection: Dragendorf's, Wagner's, and Meigs. The suitable extraction solvent for plant extract to detect the presence of alkaloid is using methanolic extract. Using methanol as solvent, and we add HCl 1% to the plant part and heating this extract in water bath for 20 minutes. After 20 minutes, we add the reagent separately, Dragendorf's, Myers, and Wallace. So the second step is analysis. The first step is preparation by extraction with methanol and addition of HCl. And the second step is analysis. Analysis means addition of the use of suitable method. In this case, we use reagents, Dragendorf, Myers, and reagents. Now, at the end of this test, we can note the change. The change involved when we add Dragendorf to the plant extract. If the colloid present, the color of the extract changes to orange, formation of orange color with precipitate. In case of Wagner, there is also change in color, formation of precipitate with brownish to red color. My reagents form creamish precipitate as end product or as end of reaction. So finally, we can report the result as positive or negative. If positive, it is present; negative, it is absent. For testing the presence of tannins in plant extracts, as we know, tannins are phenolic compounds. We use Bremer test for identification of this class of secondary metabolite. Bremer test consists of 10% ferric chloride solution as reagent. So the preparation of plant extract is by using water as solvent. We can first prepare plant extract using water, and then we separate the mark or the debris of cells from the water extract by using filtration and concentration to suitable level. The next step is analysis. We add the reagent, premier reagent of ferric chloride, 10% reagent. In interpretation, for the last step, we know the change in color. If the tannin present, the color of solution can be changed to blue, black color with formation of precipitate. Indicate the presence of tannins, formation of blue, black precipitate by using Bremer test or by addition of 10% ferric chloride solution. For unsaturated glycosides, as we know, unsaturated glycosides consists of two parts, sugar and a glycol. To test the plant extract for presence of glycoside, we have firstly prepared the extract by using water and sulfuric acid and phytic chloride. First, we add 20% few ml 20% sulfuric acid and few ml of 2% phytic chloride to what? To powder plant sample. Then boiling the water bath for 30 minutes. Note that the test for anthraquinone different from the previous test for tannins. Here we start, we are using from powdered plant material. Not we work on extract, we work from the powdered material from the start. We add sulfuric acid and ferric chloride to powdered plant material first, and we reflux it or heat it in water bath for 20 minutes. Then we're still on preparation step and extraction. We add 10 ml chloroform, the separator funnel in order to separate the aqueous layer from an aqueous layer. After shaking in separator funnel, we separate the chloroform layer, which is expected to contain anthraquinone. We take the chloroform layer and concentrate it by addition of few ml of ammonium hydroxide solution 10%. Firstly concentrated to 4 ml and we add 2.5 ml of ammonium hydroxide solution 2%, and this step it is considered as analysis step, addition of reagent. The reagent here is 10% ammonium hydroxide solution. In interpretation, the color of the solution can be changed to pink, red color, where in alkaline layer, indicating the presence of anthraquinone glycosides. Note, we have two layers after addition of ammonium hydroxide. We have already chloroform layer, which could be considered as non-polar. When we add 10% ammonium hydroxide solution, we add aqueous layer. The color changes to pink or red on alkaline layers, not in chloroform layers. That means the alkaline layer contains anthraquinone glycosides. Lastly, we report the result if it is present, positive, and if not, we write negative or minus. The fourth test in this lecture is test for free anthraquinone. That means anthraquinone are free from sugar moiety, and we expect they are hydrophobic or can be soluble in semi-polar or semi-polar solvents such as chloroform and ethyl acetate and ether, but they are insoluble in water. And you should note that also, in contrast to anthraquinone glycoside, here extraction, or we start with ethanol extract. In the case of anthraquinone glycoside, we start from the powdered sample. We add ferric chloride and sulfuric acid. Here we start with ethanol extract. We boil this extract and then we shake with chloroform in order to separate or solubilize anthraquinone in the chloroform layer. Then the next step, the chloroform layer separated and concentrated. At this level, we finished extraction and preparation of the extract for qualitative test or for general screening test. The second step is analysis step. Then we add our reagent. Also, we add ammonia solution, few ml of ammonia solution. In the last step, in interpretation, we know the change in color to pink or red color acquired by a chloroform layer now. A chloroform layer indicates the presence of free unselected. And as we expect, unselected because they are nonpolar or semipolar, can be soluble in chloroform layers, in contrast to selected glycosides, which are soluble in alkaline layer, in aqueous layer. Sorry. For flavonoid, we use Shannon test. And Shannon test consists of magnesium thermic pellets and HCl as reagents. So for preparation of flavonoid extracts for analysis, we use ethanol as solvent for extraction. After extraction and preparation, in the analysis step, we add the reagents, hydrofluoric acid and magnesium thermate or magnesium pellets. In the next step is analysis or interpretation, analysis addition of reagents. Interpretation level will know the change in color to pink or to tomato red color, indicate the presence of flavonoids. For cardiac glycoside, we use Killer-Kiliani test. As we know, cardiac glycosides consist of two parts because they are glycoside from A-glycone and sugar molecules. And the A-glycone of cardiac glycoside is steroidal compound, or consists of steroidal nucleus or mainly from 30 carbon atoms compound. For preparation of plants for cardiac glycoside test, we use methanol as solvent for extraction. After extraction, we add glacial acetic acid and 10% ferric chloride beside 6 drops of sulfuric acid. All these three reagents constitute Killer-Kiliani reagent. So Killer-Kiliani consists of three reagents: ferric chloride, glacial acetic acid, ferric chloride, and sulfuric acid concentrate. In interpretation level or step, we note the color change from green to green or blue color, indicates the presence of cardiac glycoside in the plant extract. For steroid and terpenes are biosynthesized through the same pathway of 3-methyloric acid deoxydilose pathway, and they are similar in their chemical structure. They are composed of isoprene unit. Isoprene unit is carbon 5 building block used for or as precursor for synthesis of terpenoid and steroids. Both of them, steroid and terpenes, consist of 30 carbon atoms. The test used for detection of terpenes and steroid to use Lieberman-Borchardt reaction or Lieberman-Borchardt test. This test consists of two reagents. One of them acetic anhydride, and the other one is sulfuric acid, concentrated sulfuric acid solution. And the extraction solvent used to obtain the extract by using methanol. In interpretation level, we know the change of color of the solution in test tube. When changed to blue-green color or formation of blue-green ring, this means the solution or the extract contains steroid. And if the color is changed to pink or red, pink or purple color, this means the extract contains terpenes. Saponins also are considered a terpenoidal compound biosynthesized through chemical acid pathway. But saponins have all saponins have the same physical characters. They forming froth or forming foam and when they are shaking, when we shake the water extract containing sabonins. The other important feature for sabonins, physiologically they are toxic and can produce hemolysis of RBCs. And when we will learn in next lectures, a sabonin can be tested by using RBCs because they are forming hemolysis, or the hemolysis of RBCs can be used for useful test for detection of the results. Here for sabonins, we use a frost test or foam test, and the test mainly based on using water extract. Use water as solvent to extract sample. After we extract, we shake this solvent vigorously. We shake in water extract vigorously when we note formation of persistent froth. تتكون رغوة ثابتة for a stated period of time. For this froth, then we add olive oil, few drops of olive oil, and then we shake vigorously again. We end with formation of emulsion. And this emulsion formed is indication for presence of sabonins. So the distance of sabonins is froth test and is considered as physical test. But just by addition or by shaking of water extract and addition of olive oil, a formation of froth. Also, sabonin can be tested by using thin layer chromatography or TLC. Also can be considered as cost-effective or cheap method of analysis. On TLC, sabonins can form dark bluish spot when sprayed with a spray agent, vanillin, sulfuric acid, and heating for or drying at 110 Celsius for few minutes. So TLC consists of stationary agent, stationary base, and mobile phase. Here we use silica gel as stationary base and mobile phase using chloroform, methanol, and water. While spray reagent who are vanillin, sulfuric acid. And the change or the interpretation we obtained at the end of this test will change of the color of a spot to darkish, dark bluish to black color. The third feasible test for sabonin is the use of hemolysis test. And this test based on the fact that sabonins are toxic metabolites that produce hemolysis and RBCs. The test composed of several steps. Firstly, in extraction and preparation, we use water extract and we add sodium chloride solution to this water extract. Then, the test tube, the sensor tube, we add animal blood, few drop in animal blood in the test tube, and we allow to stand for 15 minutes. So this is considered as analysis step. Addition of animal blood and addition of sodium chloride to water extract. At the end of the test, we know that the settling down of RBCs that indicate the presence of sabonin in the blood extract. The precipitation of RBCs means that they are containing sabonin compound. Fixed oil also can be detected by using TLC method, thin layer chromatography method, and in this method we use stationary base silica gel and mobile phase. Hexane is high acetate, nonpolar mobile phase or semipolar mobile phase. And we're using alcoholic potassium permanganate solution as a spray reagent. And we note the change of the color of a spot to dark brown color, indicates the presence of fatty acid in the extract. For amino acids and protein, we use the test ninhydrin test. Ninhydrin reagent consists of 2% solution of ninhydrin, and the preparation step, extraction preparation step, includes addition of boiling of crude extract, with the ninhydrin reagent for certain period of time. This is preparation and extraction step. Boiling crude extract with 2% solution of ninhydrin. In interpretation, or at the end of the test, we know the change of color to violet color, indicates the presence of amino acid and protein in the extract. For cyanogenic glycosides, this type of glycoside characterized by liberation of hydrogen cyanide gas when they are heated or they are hydrolyzed. The test for cyanogenic glycosides involves the use of sodium bichromate paper as reagent. The reagent for cyanogenic glycoside is sodium bichromate paper. The step or preparation of plant extract involves extraction of ethanol extract, by using ethanol as solvent, and we heat this extract to boil by or within or at the same time we place a paper impregnated with sodium bichromate solution, and we close the flask or the test tube with this paper. Again, for cyanogenic glycoside, we extract the solvent, the extract the plant material is ethanol solvent. Then we place the extract in test tube or in beaker. In the same test tube, we place filter paper impregnated or macerated in sodium bichromate solution, and we close these faucets and we place in water bath, heating until it's boiled. At the end of the test, we note the change of color sodium bichromate from yellow to different shades of red color, indicates the presence of cyanogenic glycoside in this extract. As I mentioned previously, the cyanogenic glycoside on heating liberates hydrogen cyanide, and hydrogen cyanide when reacted with sodium bichromate can be changed the color from yellow to red. For volatile oil considered as also terpenoidal compound consists of monoterpenes and sesquiterpenes male. Low molecular terpenoid, carbon 10 monoterpenes and carbon 15 sesquiterpenes. The test for these terpenoids, monoterpene and sesquiterpene, we use ferric chloride solution, 1% ferric chloride solution as test reagent. So for preparation of the test, we place the volatile oil in clean test tube, then we add our reagent, ferric chloride 1% to a few drops from this reagent. And then in interpretation step, we know the change in color. If the color changes to yellow, that means the extract containing or the oil containing methyl as main constituent. And if the color changes to green color, there may be two choices. If that means the crystalline may contain thymol or eugenol. All these compounds, these terpenoid compounds, give green color with ferric chloride. When the color changes to violet, that means the oil contains methyl salicylate. And if the color changes to dark blue color, that means the crystal containing vanillin as monoterpenoidal aldehyde. Another test for volatile oil, we use TLC, cellular chromatography. Also, it is favorite test, can be used in general phytochemical screening because it is simple and cheap. In volatile oil, also we use the reagent vanillin sulfuric acid, and with heating at 105 Celsius for 2 minutes. And we know the change in color of the spot to pink or brown color, or pink brown color shows that the presence of essential oil or terpenoidal compound, both monoterpenes and sesquiterpenes, both of them give pink to brown color with sulfuric acid. The stationary base for test, we use silica gel, and we use polar mobile phase, consists of methylene dichloride, chloroform, ethyl acetate, and propanol. It is a polar mobile phase. So for volatile oil, we use vanillin as reagent, and the interpretation or the presence of essential oil or terpenoid compound, the color of the spot changes to pink or brown. For phenols or other phenol, generally for phenolic compound, we use ferric chloride test or Breyer test. Breyer test. In this test, we know the change of color of the extract to bluish black color indicates the presence of phenol by addition of ferric chloride reagent or Breyer's reagent. The another test for phenol, the second test, Lieberman test. The Lieberman test, the agent consists of sodium nitrate, sulfuric acid, and sodium hydroxide. With heating the extract at the skin, we know the formation of deep red or green or blue color in the test tube. That means the extract containing phenolic compounds. The third test for phenols using choline thiocalcio test or choline thiocalcio reagent. In this test, we add 1 ml from this reagent, choline thiocalcio, and we add 20% sodium carbonate, and we know the change of color to dark blue color also indicates the presence of phenol in the extract. At the end, and as a takeaway note, general phytochemical screening is a process of identifying the presence of secondary metabolite in plant extract using feasible and cost-effective methods, and the principle of general phytochemical screening based mainly on change of color or formation of precipitates or both of them. For general phytochemical screening, there are 4 basic sequential steps to analyze the plant extract. The first step consists of extraction, preparation, analysis, and interpretation. In this lecture, we talk about the method for analysis of organic screening for 13 important class of secondary metabolites. And lastly, we report the finding for general phytochemical screening as positive or negative. Positive for present and negative for absent of secondary metabolites.